Okay, let's talk about viruses and prions, which are not viruses but have some virus like properties in the way that they can infect people, for example. So, shown here is this sort of dried out sausage looking thing is actually a bacterium. I believe that's E. coli in the center. And all around it, shaded very helpfully in false color, sort of orangey yellow, those are going to be bacteriophage, which is a kind of virus. And bacteriophage sort of feast on these bacteria. They have this particular shape because they're going to store their DNA up here in the capsid. And we think the DNA in at least some of these is under a lot of pressure. They're kind of like little hand grenades, basically. And when they dock down and land on the surface of the bacteria and bind to the right receptors, it actually opens up the protein in such a way that they will inject their DNA. It'll just come out under its own force and go right into the cell. So, pretty neat little molecular machines. Very fascinating to watch these things move around, I would say. All right. So, if we're talking about viruses, the question is, how are we going to classify these viruses? And there have been a lot of ways proposed over the years, but most of them end up not making a lot of sense. And what I mean by not making a lot of sense is that you can make a tiny change to a virus. So it's nearly identical, but it would change the host, or it would change the serology, or it would change the disease, for example. And you can have viruses with roughly the same shape, but nothing else in common. Like everything that's inside is completely different. The way they work is completely different. Disease they cause is completely different. And so none of these really gets at it. But let's go through all the possibilities, shall we? So first way, you could classify these things based on the way they look. Now the problem here is that a lot of viruses look the same. And by the same, I mean they have a shape that's called icosahedral. If you were ever into tabletop gaming back in the day, an icosahedron is going to be the shape of, for example, the 20-sided die, or actually the 12-sided die, I believe, is a different version of this. Like it's mathematically close to it. You just put basically a triangle on each of those faces. Let's see. So, classify them by how they look, you could, but you wouldn't do very well. So, poliovirus is a very small RNA virus. Herpes viruses are gigantic DNA viruses, but they both have an icosahedral core in there. And so, the core doesn't really tell you anything about the virus itself, and it also doesn't tell you anything about the disease you get. And so, you would like for viruses that are genetically related to each other to be grouped together, and this grouping by how they look does not lead to that. So no, yeah. Should you classify them by disease? Again, probably not. Yeah. For example, there are all the hepatitis viruses. Hepatitis A virus is one that you can get by contaminated drinking water, or, for example, if you were to go surfing over in California, after there's a rain, there are a fair number of homeless and or migrant families that live in the canyons, and they have to go to the bathroom in those canyons. And so when it rains, it washes all that beautiful stuff out into the ocean. And boy, some days the waves look really good, I gotta say. And so, you know what? You go out anyway. But yeah, hepatitis A is then a common thing that happens. And so hepatitis just means inflammation in the liver. And this is caused by a tiny RNA virus related to poliovirus, actually. Hepatitis B, though, is a neat one. This is an RNA virus that's going to turn into DNA and then back into RNA and then back in, no, DNA to RNA back to DNA. There we go. That's how it works. So it's one of these weird double reversing viruses. Pretty crazy. Totally unrelated to hepatitis A, like completely unrelated, but they both hit the liver. And in general, anything that hits the liver is going to be something that causes hepatitis. So even if the viruses are completely different, they can end up with more or less the same disease. Now, there are some differences in severe hepatitis B will kill you, whereas severe hepatitis A almost always won't. Yeah. Hepatitis C is going to be a chronic disease. This is one you're going to get, and it's going to stick with you for a long time. It's a totally different kind of RNA-containing virus that makes it, but it's unrelated to hepatitis A and hepatitis B virus. Hepatitis D is actually not even a virus. It's a thing called a satellite virus. And hepatitis E is going to be more closely related to hepatitis C, but actually a kind of a different branch of the family. And so there are all these hepatitises, that cause roughly similar diseases, but the viruses themselves have nothing in common. And so the answer there would be no, we should not classify viruses by disease because it doesn't tell you anything about the virus itself. It just tells you about the disease. And interestingly, for example, one of the pig coronaviruses, you can make a single nucleotide change that changes one amino acid, and it can change one of these viruses from one that causes something like a common cold in the pigs to one that causes lethal diarrhea in the pigs. And so the one case, it goes to the lungs and just stays there very happily. The other case, it only goes to the intestine. And you can put it in, and it will just go to this one place. That's the only place where it can actually grow. So, yeah, if you can make a tiny tweak and totally change the disease, then that's not a very good way to classify the viruses, because you could get two viruses that are almost the same, and you wouldn't really be able to tell what disease they cause without the full genome sequence. If that's even known. Okay, so should we classify them by host? Like, are all human viruses the same? No, that's like the dumbest one. So generally, this is definitely not the case, because humans can get viruses that have DNA or RNA that turns into DNA, or DNA that turns into RNA and back into DNA inside. And they can have totally different sets of genes that are genetically unrelated to each other, have a completely separate evolutionary history, and so classifying them together just doesn't really make sense if we're talking about virus taxonomy. And so no again. And I'm actually part of a group, full disclosure, that does virus taxonomy, that's in charge of kind of naming viruses and figuring out how they should be grouped. I'm not the head of the group, I'm just on the committee, but it's kind of neat work. And so, you can see the mess that we get into when we try to classify diseases by any of these things, by host or disease, or how they look, for example. So the way that we classify viruses is by, first of all, what's inside. So up there in the top, that's a Nobel laureate, David Baltimore, staring right through you into your soul. Look at that piercing gaze. Oh my goodness, yeah. So he came up with this thing and did not get the Nobel Prize for this, by the way. This or something else. But this way, which is called the Baltimore classification system, where you look at what the virus has inside, and based on that, and based on the genes that it contains, you can then organize these viruses. Because viruses start out from very different places. They can have DNA or RNA on the inside. The RNA can be forward or backward, or you can have both, double-stranded RNA. There are even some viruses that have a little bit of forward and a little bit of backward RNA on the same genome, like some genes that sort of point toward each other. They're going to behave more or less like these negative strand viruses. So the ones that where the RNA looks like a messenger RNA, like you could actually just translate it. It's got the codons in the right order, like A, U, G, and then something, something, something. Those are going to be called positive sense or messenger RNA sense. The ones where we're looking at the reverse complement of that. So you could not directly translate this, but if you made a copy of it, that copy would be just like a messenger RNA and you could translate it. Those are called negative strand viruses, or you can think of them as antisense viruses. And then there's DNA and everything else. So viruses basically show us that everything that life could have been during evolution. So which of these actually looks the most like us? So all these paths, first of all, are going to lead toward making proteins. That's what viruses do. That's what cells do. That's what life does. Proteins are always going to come in these viruses anyway from messenger RNA. Bacteria have a different way to make a few proteins, but almost everything in a bacterium also comes from messenger RNA. And then the messenger RNA can come from, one of a couple of sources. You can copy it from DNA, or you can copy it from double-stranded RNA. But the way you make DNA can also be different. You can start with RNA, single-stranded or double-stranded DNA, or you can start with DNA but turn it into single-stranded RNA and then turn it back into DNA to put some mutations in, basically, so that the virus can evolve a little bit faster. Or you can start with either the positive strand or the negative strand and make the other strand to get to double-strandedness, or you can just start as double-stranded and then bam, you're right there. So different paths that lead to the same goal. That's what viruses have. So which path does the rest of life take? I'll give you a minute. So what do we store our genes in when they're inside the nucleus? DNA, right? And it's not single-stranded DNA. That's bonkers. Only viruses do that. It's going to be double-stranded DNA. So we're either going to look like this or we're going to look like this. And so when we copy, when we're going to divide our cells, what do? Do we turn our DNA into single-stranded RNA, giant chromosomes of RNA, which do not exist, and then turn it back into DNA? No, because we're not like a hepatitis B virus. We take our double-stranded DNA and we just copy it into more double-stranded DNA during S phase of the cell cycle. So, yeah, and then from there we're going to make messenger RNA, and from there we're going to make proteins. And so human beings are like an adenovirus. Fantastic. Isn't that great? Adenovirus 36 is a neat one. This is an adenovirus that has been found preferentially in people that are overweight. It is strongly associated with obesity. Now, we do not know if this is a virus that you catch and it then causes some bodily changes that lead to obesity, or if this is a virus that you are more likely to catch if you already have like a predisposition toward obesity, or if you are in fact already obese. Nobody has done that trial yet, but at least as far as I'm aware. But yeah, interesting to think that, because we could totally vaccinate against adenovirus 36. Could we vaccinate against obesity? That would be pretty neat, wouldn't it? For some of us, I'm looking at myself here, that might mean more than it does to others, yeah. But yeah, I don't know. There we go. All right. So we're going to talk today about 4 different viruses. The 4 viruses, well, actually 3 viruses and 1 prion, they're going to be HIV, because I think you got to know about that one, because that's like the best studied virus in the world. We're going to talk about SARS coronavirus, but the original, not SARS coronavirus 2, although we'll mention it, yeah, because whatever, yeah, because it's trending on Twitter these days. And we will talk about a 3rd virus, which is called TT virus, torquatinovirus, or transfusion-transmitted virus. And then we'll also talk about the new variant Creutzfeldt-Jakob disease, which is a prion disease caused by the PRP-SC form of the PRP-C protein. We will get there, I promise. All right, so first of all, how do we know that HIV isn't just a conspiracy that was, hatched in a lab somewhere? Well, yeah, believe it or not, somewhere on the internet, this has been suggested. Yeah, you'd say that's pretty dumb, but whatever. There are ways to disprove things with science. Science is actually very good at that. But for years, this was kind of an open question, like where did HIV come from? What was the original origin? Because we knew about some distant viruses, and these were found in chimpanzees, and they were kind of HIV-like, but they weren't really the same as the HIV that you found in humans. The HIV in humans is 3 different types. They're called type M, type N, and type O. And the type M here is in the middle, the N is at the top, and the O is at the bottom. And what they discovered in this paper, which was published in Science back in 2006, when you were, oh my gosh, you were probably really young, yeah. This was actually discovered that chimpanzees had viruses very similar to HIV M, N, and O strains, plus a whole bunch of other chimpanzee simian immunodeficiency viruses. HIV stands for human immunodeficiency virus. So yeah, and it was only in a certain population of chimpanzees located in what is now Cameroon, and the chimpanzees' range stretches over to the Republic of Congo and a little bit in the Gabon and Equatorial Guinea as well. So yeah, that appears to be the original origin of HIV, is from chimpanzees in this area. And as for how the virus gets from chimpanzees into humans, we think this happened. So the oldest HIV samples that have been found, if I remember correctly, go back to the 1930s. And we think there was probably some evidence of the first infection happening a little bit before that, somewhere around the turn of the last century, I think 1900-ish. At that time, there was a lot of development in this area. So this area would have been called the Belgian Congo, and in the Belgian Congo, there was a brand new city, still under Belgian control back when, yeah, Europe owned Africa. Weird as that seems. um, and during this time, yeah, they had a lot of expansion. They were cutting down the forest. People were going out. And one of the problems was it was difficult to get food into the area, and so people would eat whatever's in the area. They would go out and hunt, just like you might even do. You hunt whatever is local, yeah. And so among those things, and they are actually still hunted in some of these areas, people would hunt chimpanzees, and still do to some extent. And if you have been hunting, and bear in mind that I have not, but I would imagine, again, some of you probably have, or you know somebody who has, right? But if you've been hunting and you shoot, and let's say an animal like a deer, what's going to happen next? Unless you are, you know, Rambo, crack shot, American sniper, whatever, probably you're not going to get a direct hit that is a one-shot kill. That's more for those videos they show on whatever channel that is that they play at the hairdressers all the time. Probably you're going to hit the animal and you are not going to kill it right away. And the next thing it's going to do is it's going to run off. Now, this is a very heavily forested part of Africa. There's a lot of forests that run right through the middle of Africa. And there are a lot of spiky, sticky things in the forest there, just like there are in the forests around Texarkana. So many spiny plants hanging down from trees and whatever. So if you're tearing off after potentially, you know, the thing that's going to feed you and your family for the next week, then you are going to have to go off through the forest, and you might end up with a couple little, cuts and scrapes from chasing after these things. And then once you get to the animal, you're going to have to field dress it, probably right there, because you're not going to want to drag it all the way back. You know, a gorilla weighs a lot. A chimpanzee weighs a lot. So yeah, you field dress it right there, get the meat out, and as you're doing that, it's a process where you're going to get your hands absolutely covered in chimpanzee blood. And so if you've got a cut and you are cutting open a chimpanzee or a gorilla, you're going to be exposed to any viruses that are going around in the bloodstream of that chimpanzee or gorilla. That is straight up blood to blood transfer. And so you don't have to imagine anything weird going on in the bedroom between people and zoo animals. It's almost certainly hunting that led to HIV. And the thing is, it was a very slow virus to spread. It was many years before people actually started to notice it. That was in around the 1980s that it really started to gain some attention. It was basically discovered, this thing that had been circulating for possibly something like 80 years at that point. So the other way you can kind of tell where this virus came from is with a little armchair epidemiology. So yeah. Basically, the place where you get the most different strains of the virus is probably going to be the original source of the virus, because some strains travel better than others. In this little part of Africa, which is where we think it originated, you can actually find M, N, and O type HIV. Outside of this part of Africa, it's pretty much just M type HIV. And you actually get a couple other HIV-like viruses called HTLVs, that you would also find almost exclusively in this area. So yeah, it was probably the bushmeat trade in this particular area, driven by sort of colonialism at a particular time when that was very fashionable among the European set. That is the reason why we kind of really indirectly ended up with HIV. And from there, it's just about patterns of migration. And so when people from this area went to different parts of the world, they went to the Caribbean, they went to the northern tip of South America, they went over here to parts of Southern Asia, and you can watch it over time. So you've got a little bit of the virus that starts out here, very gradually spreading out. And as the color gets darker, the number of cases are going up. And there you can see the difference from 92 to 2007. We didn't really have any good surveillance before that, so we can only speculate on what happened before, but it does line up. And there would be families, there was some migration from this area of the world to these areas around the 1970s and early 1980s. And so we think that's maybe how the virus kind of got out and got around and really got a head start on our efforts to control it. You'll also see some countries in here where, look, it looks as though there's no HIV in Saudi Arabia. That is not the case. It's just that they were a little slow to admit that there might be a possibility that people were having relations outside of wedlock and might be spreading HIV. It seemed impossible at the time, and yet they have a raging HIV problem now. It was just a more political decision not to acknowledge it at the time. All right, HIV prevalence, mostly in Africa. If we scaled countries based on the amount of HIV there, look how bloated Africa gets. Although there's a fair bit of HIV in the India-Pakistan area. Yeah, definitely. But if you look at infectious disease funding, which is mostly actually directed as HIV funding, HIV research, look at that. So basically all the countries where you have a ton of research and control money being fed in are the areas with the smallest problems, and the area with the biggest problem has probably the least amount of research. This is somewhat older data. The outlook has shifted a little bit, but this is still pretty close to the case, I would say. So yeah, that's kind of perverse and kind of bad, but also kind of the reason why we have HIV. As long as the virus is allowed to basically run rampant in people in this part of the world, it doesn't matter how much you spend on it, because it's going to keep coming over, because as people move around, as people go on vacations, and yeah, eventually it's just going to be a big problem. Look what a big problem it is in Madagascar, for example. Look how tiny Madagascar, I remember how little Madagascar should be, and look how big it is there. South Africa, too, very big problems, as are the independent states within South Africa. All right, so what does HIV do? So this little graph kind of sums it up. And this isn't data from a particular experiment. This is like a composite of all the data that people understand about HIV. So this is more a talking point graph. So in general, this is what happens. You're going to start out in a normal healthy person with about 1,000 helper T cells, which are called CD4 positive T lymphocytes. Helper T cells are like little controllers of the immune system. They are going to go out and regulate a bunch of other cells. So they'll tell macrophages who to fight. They'll tell a bunch of other white blood cells who to fight and how to fight them, and basically keep them going in the fight until it's time for the fight to end. So yeah, I think of them, CD4, I think of that as like a little 4-star general, if you like, of the immune system. And so normally you should have about 1,000 of these, and then over time, as you get HIV, the HIV is going to come up and it actually infects these cells, CD4 T lymphocytes. HIV will also infect another white blood cell population called macrophages, definitely infects those, and can get carried around on another white blood cell population called dendritic cells. We're going to learn a little bit about those in biology too. There's an immunology section of that, but for now, yeah, you don't have to worry too much about those. Just remember the CD4 T lymphocytes, and if you want to remember that other cells can be infected, that is okay as well. So there's a little spike in the virus, CD4 cells drop, but then they're going to come back, and they're going to get strong again, and they're going to push the virus down. So it's a temporary win for the virus. Problem is, when HIV infects, it doesn't just float around in the cytoplasm like most viruses do. It's going to go straight into the nucleus. It's going to cut one of the chromosomes in the nucleus, kind of at random, and it's going to insert a copy of the HIV virus genome into your genome. And so now this cell and all the progeny of this cell are going to carry embedded copies of HIV forever. And we know this is the case, and this has happened many times before, because you have about, about 4 copies of HIV in your genome for every single copy of a gene, a human gene in your genome. So you're about 4 times as much HIV-like virus as you are human, at the bare minimum. There are other ways of looking at this where you would say that you're something like 200 times as much, or maybe 50 times as much at least, virus as human. But yeah, That's something, right? A lot of this ends up as the stuff that we call junk DNA, but a lot of that junk DNA is copy after copy of HIV like viruses. And the reason why they don't kill us is that most of the HIV like viruses in our genome are actually, they have a couple of mutations put in there. There are a couple of human proteins whose only job is to try to mutate viruses like HIV out of existence so that even if they get embedded into our genome, they can't wake up again and make us sick. But in an active HIV infection, yeah, you're going to still be able to wake up at least some of these viruses. And so even if the immune system keeps clearing up the viruses, this is going to go on for years. And eventually the HIV is going to wear down the immune system. You'll have low levels all the time that'll gradually rise. And this is if you're not taking anti-HIV therapy, by the way. And then eventually your CD4 lymphocytes will crash. And when you get down below, I believe it's around 100 CD4 lymphocytes per cubic milliliter, cubic millimeter of blood, then at that point, your immune system has essentially collapsed. And at that point, you're going to catch all kinds of other diseases. So we call these opportunistic diseases. These are things that your immune system is dealing with and winning against right now. But when you don't have an immune system anymore, then any of these things that are normally very mild and very easily dealt with can become life-threatening. And you're never sure which one's going to get you, but one of them does, yeah, in the end. And it does lead up to death, yeah, in the end. So that's HIV. But the positive, and so this whole process would take years, somewhere between about 2 or 3 years to about 15 years in general. Back when there was a lot of spread and before we had the first real good treatments. Since then, though, with all the anti-HIV drugs that you can take, this is actually a very controllable disease. It's not a disease that you want to get because you're going to have to take multiple pills every day for the rest of your life to keep it down. And if you stop doing that for any reason, you're going to become infectious and able to spread the virus to other people, and then you're going to put them in the same terrible situation that you've been in. So Texarkana is, by the way, a hotspot for HIV. We punch way above our weight in terms of the number of people who are here and the number of HIV cases, HIV prevalence. So yeah, Texarkana, not doing well with HIV, even though you don't hear all that much about it. But there are clinics in town, I believe, over in the Arkansas side, and plenty of support groups around if there is somebody that you know who has this, or if you might have this. What are these opportunistic diseases? Well, here's one. It's called Kaposi's sarcoma. It's caused by a kind of herpes virus called the Kaposi's sarcoma herpes virus, KSHV, and it causes these little lesions. They're kind of like cold sores, except that you can end up with them on your arm or on your neck or on your tongue. Look at this poor guy, yeah. That looks very, very painful. And yeah, this is a disease that is normally quite rare. It's found a little bit more in people who come from the Mediterranean area. I think the Greek population has a little bit more of this disease than normal. But ordinarily, it's quite easily controlled by the immune system. And the only people that really get it are ones where the immune system has been damaged, for example, by HIV. Another thing that is just in the world normally is Pneumocystis jirovecii, and that is a little type of single-celled fungus, and it can get into your lungs, and so the top of the lungs look pretty good here. You want to see the lungs looking dark. If they look white and cloudy and stringy like these do, this means that there is fluid and mucus and cell debris floating around in there, and that is just generally a sign that you are sick. And if you have all that junk in your lungs, it means that you cannot also breathe with that same part of your lungs. And so, left untreated, this can be fatal, but it's normally very, very rare. One of these diseases that you could catch just about every day of the week, but we really only see this in patients with compromised immune systems. So diseases like HIV, where the immune system breaks down. Now, some of you, probably about 1 out of 5 people watching this lecture right now, are actually going to be not immune but resistant to HIV, which means if you were to get HIV, you could still catch it, but you would probably never progress to AIDS even if you did not take a lot of medication, and your body would naturally be much more able to control the level of HIV, so you'd have a fairly controlled infection. These are people with a modification in a protein called CCR5. Now, first of all, let's just look at CCR5. What's that look like to you? Here we got a protein that goes 1, 2, 3, 4, 5, 6, 7 times through the membrane. Is that ringing any bells? So there are 7 transmembrane helices. There would be a little part down here that you could fold up into a thing that I would call helix 8, maybe. And essentially it's going to have another protein that's associated with it called a G protein with alpha, beta, and gamma subunits. Yeah. Ring a bell yet? Right. This is a G protein coupled receptor. CCR5 is one of your GPCRs, and it's involved in communication between different cells of the immune system. But this is also the protein that HIV is trying to grab onto. So HIV is going to use CD4 to get close to a cell and attach to a cell. It's just an attachment receptor or a co-receptor. And then what it actually is looking for is this CCR5 protein, or there are some cases where HIV can get in through CCR2, I believe, protein as well. And so, yeah, the virus is trying to bind this. But roughly 1 out of 5 people who grow up in the United States, and this is people of all whatever, ethnicity or social group or anything, are going to have a thing called the Delta 32 mutation, which means you're going to be missing 32 amino acids, which is most of helix 5. And you remember that helix 5 is pretty important. If you don't have helix 5 there, you cannot signal with your G protein coupled receptor, because it's 5, 6, and 8 that are going to do most of the work, right? Yeah, people with the Delta 32 mutation actually make a broken version of CCR5. It does not get to the surface. They are much more resistant to HIV infection and HIV spread once it does infect. Yeah, they will control the virus that way. So that's neat. And yeah, you could do a pretty quick little PCR test and actually read out whether or not you have CCR5 deletion in your particular copy of the genome, or whether you have 1 copy or 2 copies of the Delta 32 mutation. All right, what's HIV look like inside? Well, it's got 3 big genes and then a couple of other little tiny genes that are kind of here and there. They aren't in all strains. You got HIV down here, looking great with the green, red, and blue genes. The green is called the group antigen gene, or GAG gene. It's going to make the shell of the virus. And this thing looks like a sawed-off ice cream cone in the center, which is going to protect the 2 copies of the virus genome. So the virus has 2 single-stranded RNA genomes inside, 2 copies of the same genome. So the bad side of that is that if you get infected with say you pick up a resistance mutation in one of these and you pick up a different resistance mutation in another one, if you get both of those packed inside the same shell and they then infect, then you are going to start out with doubly resistant HIV. And that's going to be very hard to treat, yeah. Let's see, they also have the envelope gene, which is going to make the protein that sticks out on the outside. That's the one that's going to grab onto the cell and let the virus in, equivalent to the spike protein of something like a coronavirus, if that's a more familiar one. And then you're going to have the Pol section, the polymerase, and that's going to include all the genes that copy the virus in there, and the ones that sort of cut the chromosome of the host and glue the copy of the virus into that chromosome. So the virus is going to start out as viral RNA. It's going to be converted to DNA by reverse transcriptase, integrated by the integrase, and then turned back into RNA by cellular enzymes. So we left out a couple words right in there, but you've got them now. Beautiful thing about HIV, if it can be said to be nice in any way, is that at least we've got options for treatment. So at every step of the HIV life cycle, there are generally multiple different drugs that are FDA approved that you can actually take. And most HIV treatments, modern HIV treatments, are going to involve at least 3 of these drugs at the same time. It's called combination therapy, or highly active antiretroviral therapy, HAART. And yeah, that's the way we treat HIV. Basically, you hold it down from different directions. Each of these things beats on the virus in a different way and basically helps your immune system to hold back the virus. So HIV can be treated. HIV cannot ever be cured, of course, because HIV, the first thing it did was put a copy of the HIV genome into your chromosomes of every single cell that got infected. And so, yeah, your cells are now preloaded with HIV virus. And it's eventually going to come back. And so that's why you have to take some drugs like these for the rest of your life. All right. So here's some of our information. This is most of the stuff you need. Once again, as in the histone chapter, I would say you're going to want to make yourself a little table where you keep all this information. So what does HIV stand for? It's human immunodeficiency virus type 1. What's the genome of HIV? It's 2 copies of single-stranded RNA. So put like a little 2X ssRNA there. They're going to be replicated via a DNA intermediate. The structure of the virus is going to be enveloped. So it's going to have a membrane around the outside with proteins sticking out of the membrane. The membrane is going to look just like the plasma membrane of the cell, and that's actually what it is. The virus is basically ripping off a piece of the plasma membrane and taking it with it, like a serial killer wearing the face of a victim around like they do in the movies. Yeah, disgusting. But viruses are terrible. Never forget that. The disease associated with HIV is going to be AIDS, which is acquired immunodeficiency syndrome. And the treatment is going to be, there are many different kinds of treatment. Lots of antivirals that target the reverse transcriptase, protease, integrase, and the fusion process. Those are the most common ones. There is currently no vaccine available. Several HIV vaccines have been attempted. None of them has actually worked, unfortunately, so far. So a lot of clever ideas that didn't really go anywhere. The virus is going to bind to its receptor. We've left that off of here, but remember the receptor is actually going to be CCR5, and the co-receptor is going to be CD4. So that's CCR5, not CR5, like it says in the text below. And the origin, that's the last thing we've got to remember for each of these viruses. The origin is going to be in non-human primates. We believe chimpanzees are the origin for M and N types, and gorillas seem to be the origin for O-type HIV, which does not spread very well from human to human. And then from that point, it's transmitted by humans to other humans. Mostly it's a sexually transmitted disease, but you can also catch it via transfusion or anything that would mix blood with other blood. The virus basically doesn't have a way out of the body except through blood and some bodily fluids. And it doesn't really have a way into the body without some kind of little cut or scratch that would give it access to, again, the bloodstream. All right, so done with HIV. Let's now talk about one of my favorite viruses, which is a coronavirus, like the SARS coronavirus. So you can see it's over here on this side of the chart. It's going to be a positive sense RNA virus. So basically, the SARS coronavirus looks like a giant messenger RNA. And by that, I mean it has a cap at the 5' end, which messenger RNAs have. And it has a poly A tail at the 3' end, which messenger RNAs have. It's going to replicate by copying that into double-stranded RNA. From that point, it's going to make messenger RNAs, and once it's got messenger RNAs, they're going to go to make proteins, and yeah, that's the story of SARS coronavirus right there. So this is what SARS coronavirus looks like, and this would actually be true for SARS-1 or SARS-2. We're showing chest x-rays from SARS-1 here. So on the left, we've got a patient that actually had fatal SARS coronavirus. And as you can see here, look at all that infilling. Look at all that white stuff up in the lungs. This person basically has about 1/3 of one lung available for breathing. And despite being on a mechanical ventilator, this person succumbed to SARS. Yeah, because basically you can't breathe with, 1/3 of one lung. That's why you got both of them there. This is a happier case. This is a person that actually survived. And so here you can see about 2 weeks after infection, you've got some filling in here. So it's a very slow process, as is SARS coronavirus 2. It takes something like a month between initial infection and when you would die. And it takes something like 1 to 2 weeks before you would notice symptoms. So we can assume this is somewhere between 1 and 2 weeks from the point of infection. Here you've got a lot of filling in the lower lung. So it causes a lower respiratory disease. And here we are at kind of the peak of the disease. See, it's on the 21st of January, February, March, April, May. There we go. We know our month numbers. And you can see here at this point it was very dire, and this person was on a mechanical ventilator. They've got less than half of their normal lung capacity available. And then here you can see the resolution. So another week later, and it's gotten a little bit better, but you still have a lot of scar tissue there. And a further week later, so at this point the person was virus negative, was able to be discharged, but look at all that scar tissue down in the lungs. Some of this stuff can take months to clear out, and it is the same in large part in SARS coronavirus 2 as well. There's damage in there. We also know that in SARS coronavirus 1 and 2, the disease causes a lot of damage to blood vessels and potentially the heart. So quite often it's something like a stroke or an aneurysm that would actually kill people in this disease if they don't just run out of oxygen first. So yeah, very tricky if you are a doctor or a nurse, somebody involved in the hospital, in keeping coronavirus patients alive with one of the SARS coronaviruses anyhow. All right, what is the disease in SARS coronavirus 1 or 2? It's basically the same disease because they're basically the same virus. It's called an uncontrolled exuberant immune response, or for short, a cytokine storm. So cytokines are like little messages. They're little tiny proteins that can be released by different white blood cells. Among the white blood cells that can both release and respond to cytokines are macrophages. And so in SARS coronavirus 1 and 2, and this graphic is actually from bird flu, and also in Ebola, you get a situation where your macrophages can get infected. And when your macrophages that are either in the surface of your lung or in your bloodstream, depending on which disease we're talking about, when those get infected, then they're going to release all their cytokines at once. And the cytokines will do a couple of things. They will damage, just non-specifically damage, all the cells in the vicinity, and they will call in more macrophages, which will become infected and activated, and will cause more damage that will call in more macrophages until you basically run out of macrophages or die from all the damage that the macrophages are causing. In a disease like SARS coronavirus or SARS coronavirus 2, this is going to happen on the surface of the lungs, but there are a lot of blood vessels in the lungs. So some of the virus isn't going to get into the bloodstream, but some of the macrophages that are activated will be in the bloodstream, and so they're going to be running around the bloodstream and causing a lot of damage to the blood vessels. And also causing some coagulation, some little blood clots right there in the blood vessels, which can then end up lodging in the heart or in the brain or down in the toes. That's what COVID toes are all about. It's little tiny strokes down in your toes, basically, little blood clots. Yeah. And the cytokine storm is going to be driven by a number of proteins like tumor necrosis factor, macrophage inflammatory protein 1 alpha called MIP1 alpha, and interleukin 1 beta. In SARS coronavirus 2, this process is going to be driven a little bit more by a thing called interleukin 6. And as a result, we're going to make things like nitric oxide, and some of these cells can actually make hypochlorous acid, which is the same compound you would find in bleach. It's actually the thing in bleach that kills bacteria. So, yeah. This is pretty strong medicine, and when macrophages release all of it at the same time, then it causes a very strong immune response, and there's basically no way to shut off this immune response. It's very difficult to. And so the virus doesn't kill you right away. It sort of builds up. It causes more damage and brings in more macrophages every day over the course of about a month, or maybe a little more than a month, until eventually something breaks down and it causes death. That is essentially the disease in SARS coronavirus 2. So it's this kind of cytokine storm. So here's a look at SARS coronavirus 2 or SARS coronavirus 1. They are, again, genetically almost identical. They have all the same genes in all the same places. Each of these has somewhere in the vicinity of 30 genes. Down in this area, there are a couple of genes that we're not completely sure if they are made or not made, but it looks like something that could be a gene, and so that's the uncertainty there. But you can see in terms of conservation, the ones that tend to be most similar from one coronavirus to another are going to be these that are right here, and these are the polymerase and a couple of associated proteins that work with the polymerase. So the things that tend to be conserved among coronaviruses are the genes that copy the coronaviruses. And then, to make a coronavirus shell, you're going to put together about 2,000 N proteins, about 1,500 M proteins, or membrane proteins is what that stands for, just a handful, maybe 5 or 10 envelope proteins, or E proteins, and you'll have about 240 spike proteins that are going to be bundled together in groups of 3 that'll make somewhere up to about 80 spikes on the surface of the virus. So S, E, M, and N, those are the 4 proteins that make the shell of a coronavirus. Now, coronaviruses come to us from bats originally. So we know this because there are a lot of relatives of SARS coronavirus that are found in bats. And these are found all over Asia, Southern Asia in particular, and they're also found on the east coast of Africa, around Kenya. There's a population of bats that has SARS-like viruses in them all the time. These bats seem to carry the SARS-like viruses for life, and they don't seem to suffer any ill consequences from them. It's just when the viruses get into humans that they cause very bad problems. These are some of the bats that carried SARS coronavirus 1, Rhinolophus bats. SARS coronavirus 2, the closest relative of this virus, is carried by another Rhinolophus bat called Rhinolophus affinis. Rhinolophus bats are known as horseshoe bats because they got this little thing on their nose that kind of looks like a horseshoe shape. So they're pretty adorable, actually. All right, so SARS coronavirus stands for Severe Acute Respiratory Syndrome related coronavirus. Its genome is around 30,000 bases of single-stranded positive sense messenger RNA with a cap and a poly-A tail. It has an envelope, just like HIV does, which it steals from the cell that it last was in. The disease associated. For SARS coronavirus 1, it is SARS, Severe Acute Respiratory Syndrome. For SARS coronavirus 2, we call the disease COVID-19, Coronavirus Disease 2019. In both cases, the virus is going to enter cells through the receptor, which is called ACE2. And so there it is, ACE2. And ACE2, it's going to have a little help from other proteins, but we'll just focus on ACE2 receptor. I think that's enough here. And both of these diseases are going to cause lower respiratory pneumonia with extensive macrophage-induced cell damage that can lead to blood clotting diseases, including strokes, and also to aneurysms. As for treatments, now we can you change this. Yeah, so in SARS coronavirus 1, we actually didn't come up with a treatment. We just managed to isolate the people, and they either got better or didn't, but we managed to stop the virus from spreading back in late 2002, most of 2003, and then it came back a little bit in 2004. But now we have remdesivir, which actually works and holds down the number of viruses. Can't necessarily save your life. It's not strong enough to do that. It can't turn off the cytokine storm, which is what that disease actually is. But it helps. It's going to help the immune system, and it can get people out of the hospital a little bit faster than they would originally. There is currently no vaccine, although people are working on vaccines for SARS coronavirus 2. And so, yeah, if they've got a vaccine and it's licensed and works by the time of the test, then that's going to be an acceptable answer for the test. But as of right now, when I'm talking, there is no proven licensed vaccine for either of the SARS coronavirus strains. And we believe SARS coronavirus originated in bats. The original SARS coronavirus was then transmitted to an animal called the Himalayan palm civet, which is a food animal that's farmed in China, and from there to humans. We think SARS coronavirus 2 is in bats. It may or may not have gone through pangolins, or they may just have a very similar set of viruses to the ones that are in bats. And once again, it ended up in humans that are then spreading it amongst themselves. All right, next step. So this is the Baltimore classification system. And once again, let's look at a third virus. So we covered a double-stranded DNA virus. We covered a single-stranded positive sense RNA virus. Now let's cover a single-stranded DNA virus. And there it is, TT virus. Yeah, let's talk about those. So, shown up at the top, this is still, I think, the best picture we have of TT virus, which is kind of crazy. This virus has two names, or you can say it has no name, yeah. But for the purpose of the test, let's say it has two names. This was independently discovered at around the same time by two different groups. One was in Japan, the other was in the UK, over in Europe. And one group called it Torquetino virus, and you'll still see that in the literature sometimes. And the other group, which I think was actually just after the ones that named it as Torquetino virus, called it transfusion transmitted virus. But the cool thing there is that if you take the initials, either one of these is TT virus. And so a lot of people will call it TT virus, and I think that is actually the correct name for it, but you will see it described as Torquetino virus sometimes. It's related to a thing called a circovirus in pigs, and the circovirus can cause lethal diarrhea. So there we go. That's wonderful. Now, the other neat thing about TTV is that you almost certainly have it. TTV only infects humans, and some kinds of primates have related viruses. Yeah, we know you're infected because in tests that have been done around the world, when they look at healthy people with no illness to speak of, it turns out that they have a whole lot of virus inside of them. So this is a study from Russia where they looked at people, and the average person had somewhere between 8 times 10 to the 5th and 3 times 10 to the 6th. So about 1 million copies of virus per milliliter of their blood. And that's a lot of virus. That's, yeah. That's like a raging case of Ebola. That's more virus than you would have in your body with most cases of HIV or hepatitis C virus. It's really a lot. And so, yeah, you almost certainly have this right now. You didn't have it as a baby, though. And we know this because they've gone out and tested babies. And so in a couple of tests, and the early tests, they weren't very good at the actually doing the test, and so they figured that something like 50% of people ended up with this. But in the more recent tests that they've done, it actually gets up to, by the time you're about, looks like 5 to 10 years old, it's pretty close to 100% of people will be TTV positive. We think you catch this virus from either contaminated food or contaminated drinking water. It's probably going to be fecal to oral contamination, so somewhere along the way somebody didn't wash their hands after they went to the bathroom and then prepared food, or it got into the water supply and was so small that it didn't get filtered out and yeah, went over. As for how much virus you have, look at this. So this is another one of these studies in babies where they showed that nearly every child is infected by 10 years of age, but this study only looked at little babies up to 1 year old. And when they first came out, none of these babies were infected. But by the time they were 2 months old, look at that, the average amount of virus is about 10 to the 4th copies per milliliter. And by the time they're a year old, they've got somewhere north of 10 to the 5th copies of TTV per milliliter in their blood, in their entire body. Which, that's a lot of blood, right? A lot of virus. So, TTV, yeah, the scourge of humanity. When I heard some of the people that discovered this talking, they said that TTV may be the only 100% fatal virus in the world, because every other virus there are at least some survivors of. But TTV seems to infect roughly 100% of humans, and 100% of humans eventually go on to die. So there's no real way to prove that TTV doesn't kill everybody, except that it almost certainly does not. So TTV is not actually associated with any real diseases. And if you look at people that have low numbers versus high numbers of TTV, it is very hard to find any difference in their health condition. So it doesn't look as though it plays a big role, but it's hard to say because everybody's got it. So the genome is a little circle of DNA that looks like this. Here are some examples of, this is the main TTV, and these are different related viruses. See, we've got a big gene right over here that's going to copy the virus. Then we've got a couple little genes that are going to make the shell around the virus, including 3 little genes that are kind of piled up on top of each other over at that end of the genome. So very efficient virus. Here's what it does. So if you put TTV into a cell culture, like cells that you're growing in a laboratory that have either liver cells, hepatocytes, or monocytes, which are a white blood cell that will eventually turn into macrophages. What it does is it will kill a few of these cells eventually, but very slowly. So we've got nice healthy cells that are here that are stuck down. You can tell they're healthy when they look sort of like long triangles or long, cells like that. That's what happy cells look like. And when they start to get sick, they go round like a little ball, and they're actually going to lift up off the surface. So you'll see these little blobs of dying cells over here. And so here are normal liver cells down here. And here are infected cells, early, middle, and late infection. You can just see more sick cells there. So this virus does actually kill some cells. So we would certainly be healthier and probably a little happier without the virus. But yeah, in terms of actual disease, it doesn't do a whole lot. So it's been associated with a small increase in the severity of diseases in which there's a lot of inflammation, for example. We think that the DNA of the virus itself sets off an pro-inflammatory cytokine production. So kind of like the cytokine storm, but at a much, much lower level in infected cells. And we know that this virus does hit both macrophages in the bloodstream, like in the cytokine storm, and it also hits liver cells in the liver, hepatocytes. Yeah. But it doesn't do much. The best thing that I've seen, or the strongest evidence so far, is that if you already have hepatitis B and hepatitis delta, so multiple hepatitis, and you have a high TTV load at the same time, then in general you will end up with more liver damage than if you have those two other viruses but a low TTV load. So it can cause a little bit of additional misery on top of other diseases. So yeah, TTV can stand for TT virus, transfusion transmitted virus, or torquetinovirus. It is circular double-stranded DNA, circular single-stranded DNA, genome. And this one just has a protein shell on the outside. So this is one of those icosahedron-shaped viruses. So it does not have an envelope. It does have a protein shell, is what that's indicating there. And the DNA is going to be inside the protein shell. The disease associated is just a small increase in liver damage with multiple hepatitis. That's the most compelling evidence. We do not know the receptor for this virus. We do not know the origin of this virus, although we think it's restricted to humans and other primates. So this may be another virus that's come over from primate to human, or it could be human to primate, honestly. We do not know that for sure. There's no treatment for this virus, even though you have it right now, and there's definitely no vaccine for this virus, although I can see potentially a use for a vaccine for this virus, although getting kids to take vaccines, or rather getting their parents to give them vaccines against viruses that will actually kill them is hard enough in this day and age. I don't know that marketing a vaccine for a virus that probably won't kill you but might make your life a little worse would necessarily find a big enough market. But it's something that definitely could be done. Okay, so that's our 3 viruses done, and now the last thing is to look at prions. So prions are not viruses, but they can kind of self-replicate. A prion is kind of like a bad idea. So it's a protein that you already have that's actually part of a healthy you, but when the protein gets misfolded, then it causes a disease. So kind of like in Alzheimer's disease, you can have these proteins and they can misfold, and they can just build up these Alzheimer's plaques that end up causing a lot of inflammation and just lead to bad stuff happening. Prions were not discovered, but the Nobel Prize for work on prions did go to Stanley Prusiner, who was, this is a very young Stanley Prusiner. When I got to hear him talk a couple of years ago, he was about twice the man that you see there, if you know what I mean. Good eating, good living. Yeah, he'll do that to you. Yeah. Let's see. So these are proteins that can change shape, and when they're in the disease-causing shape, they can bump into a normal shaped protein, and they can cause it to change its shape. So it's like a chain reaction, shape-changing protein. Yeah, isn't that neat? I think it is. And prions are a thing that you can contract by eating prion-contaminated meat. that has the disease-causing form of the prion in it. So let's talk about these. First of all, the prion protein is actually good for you. This is needed in your brain. It binds to something, and we actually, weirdly, don't know what the receptor is for the prion protein. Figure it's probably some kind of, probably GPCR. We know it is involved in the brain because that's where the prion protein is expressed, and so it would make sense that whatever it is interacting with is also up there. But still weirdly, this is a thing that is not known, despite years and years of people dying from prion diseases. The normal type of the prion protein, which is shown here at the bottom, this thing is called the wild type, means normal type. This is called the prion protein cellular form, so PRPC. The C is the good version. This is the version that's going to keep you healthy, basically. So the current thought is that the prion protein, whatever it's doing, is binding to some other protein and causing some kind of signal to happen via binding to that protein. Because when you take away, pardon me, the protein, you still get a little bit of whatever that signaling is, but you do get some pathology. So in knockout mice, they have learning difficulties. They have a harder time learning to run mazes, for example, or doing other tests of mouse intellect. Yeah. Yeah, if you can imagine such things, which mouse scientists definitely can. But you can make different versions of this where you delete certain parts of the prion protein that will then cause severe pathology. And among the severe pathologies, they will sometimes cause neurons to demyelinate, which is the myelin is the protective insulating sheath around the neurons that keeps the charge from jumping just randomly from one neuron to another one. Basically keeps the electrics in your brain working properly. And yeah, when you lose that, you get something that's kind of like Alzheimer's disease, or kind of like an epileptic seizure, where you just get neurons that no longer function. Eventually, what this looks like is that it will cause really rapid degenerative brain disease that will eventually lead to paralysis, and then you'll lose the neurons that actually keep the heart beating and keep the lungs functioning. And at that point, people usually die, but there's a very severe dementia that's going to come along there. So they may not mentally have a whole lot left at the point at which everything else gets knocked out. But yeah, pretty terrible disease as they go. Okay, and prion diseases are not something new. This is something we've known about. And one of the more interesting prion diseases is called Kuru. So that's spelled K-U-R-U. And Kuru is something that used to be found in Papua New Guinea. And this was only found in the Central Highlands there. And this was common among people and was actually associated with cannibalism. So somebody got this disease form of the prion protein, which is called the prion protein scrapie form, PRPSC. And then there was sort of ritual cannibalism. When someone would die or when they would kill someone, then it was customary to eat the brain to sort of take on the knowledge and power of the victim, I think is the explanation. And yeah, they used to have tens to even something like 100 cases a year. Universally fatal. We don't think there are any cases of this where the person didn't die. But yeah, definitely don't eat brains, kids. Stay in school. Because brains have neurons. Neurons have lots of prion protein. And if you have a defective prion protein, that is exactly where you're going to find it. There are also other diseases associated with this. So the form that you get from eating tainted beef in general is going to be called new variant Jakob-Creutzfeldt disease or Creutzfeldt-Jakob disease. You can put those in either order. So new variant CJD or JCD, or the one from eating brains, that's specifically called Kuru. But yeah, from eating tainted beef. This is something that went around particularly in the UK, although there were some cases in the US. And this is this comes from eating cattle that are infected. Now, some of these cattle were what they called downer cattle, cattle that could not walk on their own into the slaughterhouse. And so normally the rules are such that you are forbidden from processing those for meat because if they can't walk in, it means there is something wrong with their brains, and one of those somethings is they may have prion disease, or they've got some kind of debilitating disease. And so we just say, no, we do not want that meat to enter the human food chain. But, some cows may be able to walk just enough to get in, or some investigators may be, regulators may be looking the other way, and occasionally one of these downer cattle gets into the food system and can then cause disease in humans. Now, how did the cows get the disease? I think that's another interesting thing. So the cattle get it from eating either other cattle or sheep. And if you know anything about cattle, you would say, no, I remember that cows mostly eat grass, right? Or corn. They're happy enough with corn, or give them some nice alfalfa hay or something like that. They're okay with that. But one thing that is done in some agriculture is that you will give sort of boosters to cattle. And so there are these little mixes that people would mix in with some of the feed for commercial livestock operations. And one of the mixes is actually a little bit of bone meal. And if the bone meal is not prepared properly, some of this can come from infected sheep or infected cows. And unless you absolutely prepare that properly and just about incinerate it, there will be a chance that prion protein will survive, because it forms a shape that is nearly indestructible, and eating just a small amount of that can lead to prion protein disease. So yeah, that's fairly terrible and difficult to spot. So cows eating other cows. Sheep also can develop this from the same causes. Currently, in most parts of the world, the contaminated batches of animal feed have been banned. You're not allowed to feed them to sheep or cattle. But you are still allowed to sell that feed made with downer cattle for use in feeding chickens and in feeding fish. So, yeah, we're still using it out there, and so far there's no indication that either of those animals has a prion protein that's capable of reacting and causing the prion disease. But, then again, you'd say this is how we got into this problem in the first place, and maybe it would be better if we didn't do that. But it's a, market efficiency kind of thing. It's potential food going to waste. And, the market abhors a vacuum, right? So shown on the left, this is the structure of the normal prion protein. It's the PRPC, the healthy form or cellular form, prion protein cellular form. The prion disease in sheep is called scrapie because they will have this sort of wiry coat and will end up just rubbing themselves weirdly against other things and kind of scraping themselves against the fence posts, as their brains start to deteriorate. It causes a similar disease. And they die of neurological disease complications. But look how it can change. And so it goes from, here's our little alpha helix, and then long alpha helix, and then two little strands of a beta sheet, to long alpha helix, just like normal, shorter alpha helix over here, but all the rest of this protein ends up folded into, they're red, so you can tell they are beta strands, but they're folded up into a helix of beta strands. So this is one of the very rare times when we have a beta helix. It's a beta strand organized into a helix instead of a beta sheet. Yeah. Or you can think of it as a triangular beta sheet. Triangular prism beta sheet. This form ends up being kind of inaccessible. Normally we get rid of proteins by cutting them with proteases, but whereas you have lots of nice loops on this protein where you can just get a protease in there and cut it apart, in this one, most of those loops are tied up in this beta sheet. And so the beta sheet doesn't end up getting cut, and it's the beta sheet part that's going to interact with other beta strands, on other healthy prion proteins. And it will convert the cellular form into the scrapie form. So it's kind of meta. It's like a bad idea. You've already got the protein in you. The protein is fine inside of you. But if your protein bumps into one of these misfolded scrapie form versions of the protein, even if it's from a cow, you will get the your proteins converting into the scrapie form of the protein. So, Definitely not a virus because it's just a protein. It does not have a genome at all. It's just a protein that bumps into other proteins and changes their shape. But some of the features about it actually made it seem originally like it's a virus. So when Prusner was investigating this, he came from the field of virology, and he thought he was working on a virus, just trying to figure out what this was and why it caused such a terrible disease. And then later went on to do a lot of experiments that ended up actually proving that it was not a virus. There was no DNA or RNA associated with this particular virus. It's just a protein. And protein was enough to transmit the infection. And a lot of that work was done in yeast. But there are different strains of the protein that actually cause different diseases. And so these strains end up being slightly differently folded. So with generally a longer or shorter version of the beta helix there. And that strain will keep its characteristics. So if some cause really rapid onset Alzheimer's-like disease and others cause, ruffled fur and scraping their brains out kind of disease, then they will keep that with those strains. So different neurological effects. And you can type the different strains. There's a very strong bacterial protease called proteinase K, and it can cut these proteins exactly one time. So the uncut proteins at the top, and then these are the two cut parts of it. And you can end up with different size fragments, and the different size fragments is what tells you you've got different strains there. And again, we're just looking at different size protected bits, which are that beta helix. The other interesting features here is that if you take one and you passage it in the same species over and over again, you'll actually get adaptation. So the first time it may take you 2 years to go from inoculation to disease. But if you take out a little bit from that animal's brain and you feed it to the second animal, it will then be shorter time. It may be 1 year the second time. You may get it down to 6 months the third time. So adaptation somehow, in a way that we don't really understand, to different animals. You get some cases where you have a species barrier, where certain species can transmit to other species, but not to even other members of their own species, other breeds. Like there's certain breeds of sheep that cannot catch scrapie from other ones. Sheep can pass scrapie to certain kinds of cattle, but humans eating mutton, lamb meat, cannot catch scrapie directly from the sheep. We can only catch it from the cattle or from eating human brains, which thankfully does not happen very much anymore. But you will get also some species where they will have the prion in them, and even before they actually have disease, they can still, if you were to grind up their brain, transmit it to other animals. Now, if you were to allow these to go on longer, they would eventually come down with the disease. But yeah, this means that, at least in some cases, you could potentially catch this from cattle that are not yet downer cattle, that have the spark, the little bit of prion protein scrapie form that is not yet turned into the full-blown disease. So, variation, adaptation, different classifications, species barriers, asymptomatic carriers. These are all things that we would normally associate with viruses. In this case, a prion is still definitely not a virus, but it's a little bit virus-like in the way it does business. So that's why we're talking about them here, and because it's a neat topic, and I don't know where we would fit this in otherwise. Yeah, so prion stands for protein-only infectious agent, and it's the PRION. Or the PRION. It's kind of a terrible gerrymandered portmanteau, I guess, sort of an abbreviation from different parts of words. But yeah, prions, there you go. This is a healthy brain. This is a brain with prion disease in it. And you can see how shrunken and small it looks. And if you were to look under a microscope, what you'd see would be a lot of tangles in there, instead of nice, healthy-looking cells. It actually looks a lot like Alzheimer's, except that it comes on very strong, very fast. Usually it is a year or less from the point when somebody starts showing symptoms and can be diagnosed as positive until the point where they are dead. Yeah. There is no treatment for it. It has no genome. The sheep gives it to the cow. The cow can give it to another cow by cow cannibalism through farming. And humans can get it from either cows or from other humans by consuming brains, and that's the Kuru form. The disease is new variant Jakob-Creutzfeldt disease, or Creutzfeldt-Jakob disease if you're feeling spicy. And the receptor is going to be the prion protein cellular version. And the disease-causing organism is actually just the misfolded version, which is the prion protein scrapie form that actually gets in there and causes the disease. So, there we go. That's the same thing. We're going to ask the same questions about prions we were asking about all the other viruses, and that's how you answer them. So, you've got a little work to do. You need to go through and build up that table. We need to know what is the... so for each abbreviation, which I'll give you, I'll say prion, you're going to tell me, okay, that stands for what's the name? So spell out the entire name. You're going to tell me what's the receptor? How does that get into cells? You're going to tell me what does it have for a genome? So in the case of prions, they don't have DNA or RNA. They have a prion protein scrapie form, which has a beta helix structure in it. You're going to tell me what kind of disease they cause. In this case, it's going to be new variant Creutzfeldt-Jakob disease, or you can say Kuru, that's fine. You're going to tell me how one catches these diseases. And so in the case of, what was the transmission chain before we started going human to human? And so it's sheep to cow to cow, and then cow to human, or human to human via cannibalism. And we don't know how the human got it in the first place, presumably from some version of livestock. There are deer prions that you can find in deer across the U.S. So far, they do not appear to be transmitted to humans. But this is something that I would say we need to watch. And they're going to tend to be transmitted in neurons, in deer as well as in anything else. So I would avoid the brains of deer if you have the option. If you are out hunting, it's potentially a risk, but so far it's not a risk that has made anybody sick, as far as we know. Last thing is going to be, are there any treatments available for this? And in the case of prions, as in the case of so many of these, there are no treatments available. But for HIV, remember, we have lots of treatments. Things like AZT is one of those treatments. And for SARS coronavirus, we have remdesivir that works well against SARS coronavirus 1 or 2. All right, there we go. I think that was a lot, and I hope that was interesting. This is just a little bit of sort of infectious disease science, which is something that is probably important in the world right now, maybe a little more so than it has been in the past, but it's something that kind of comes and goes. There are a lot of infectious diseases in the world, and far more infectious diseases than there are people that work on them. So, the world needs virologists. The world needs other infectious disease specialists. So think about that as you're picking careers, I would say. And as you're picking upper division modules, we do have a virology course that you can take your junior or senior year if you would like, as well as some other medical microbiology and human pathology survey of human disease courses. All right, for now, that's the end of this lecture. Thanks very much. And stay safe, stay healthy.